Hi, this is Dr. Neela Bakore, and in this segment we are going to start with cell cycle and cell division. So let us take cell cycle first. Cell cycle involves all those cyclic changes which take place in a cell when it has to prepare for division and when it divides. So cell cycle is actually an event, or rather a chain of events which takes place. As it is a cycle, that means it is going to be a cyclic event. It is going to end at the point where it starts. Cell cycle is divided into two parts. First part is known as interphase, and the second one is known as M phase, M or mitotic phase. In interphase, we find three subphases. First subphase is known as G1 phase. G here stands for gap, so it is also known as gap 1 phase. Actually, during this phase growth takes place, so it is also known as a growth phase, but G does not stand for growth here, it stands for gap. The next subphase is known as S phase. S stands for synthesis or synthetic phase. And third subphase is G2, that is gap 2. Again, during this there would be some growth, accumulation of substances would take place, that we will see in a minute. So this is gap 2 phase. So there are three subphases in interphase, and this interphase would be followed by M phase or mitotic phase. As it is a cyclic thing, we will draw it in a circular manner. Suppose this is the point from where the cell is going to enter the cell cycle. This is a newly formed cell and now it has to divide one more time. So how would it undergo changes? How would division take place? Those are the things that we are talking of. As it is cycle, we are again drawing it in a circular manner so that start and end points, they remain the same. Now this cell, it is going to enter the first subphase of interphase, that is G1 phase. So from here, whatever takes place up till this point would be termed as G1. What all things take place during this G1? It is gap 1. Now whatever changes take place actually prepare the cell for the next phase. The next phase here is S phase. So this will be followed by, so after this all those events which take place will be during S phase. So this is S. After this the cell will enter the gap 2. That is, this would be G2 phase, and the shorter duration period, only this much part, is when the cell is actually going to divide. So this is M phase. So this is how we are talking of. That means starting from this point, if you start from here and we complete G1, S and up till here G2. What is happening up till this point is all interphase. So this is which includes G1, S and G2. G1, S and G2. So in a cell cycle, you can say the maximum part goes for the preparation, and M is the division thing where the cell is actually going to divide. this interface, people think that there is nothing which is going on inside. So sometimes, earlier it was called a resting period. It is a misnomer. It is not a resting period. So many things are happening. There is growth in G1, then DNA replication will take place, again growth will take place. So all these changes are taking place. Only thing is we don't see anything from outside. So it was believed that this was a resting period, but now we know that it is not resting period, it is a period of activities which are not visible from outside. Now let us see what all happens in these phases. In G1, the cell has to prepare for S phase. In S phase, DNA replication will take place. So whatever is required for DNA replication, it will take place in G1. What should happen? Protein synthesis. What is required for protein synthesis? RNA. So RNA synthesis takes place in G1. Accumulation of proteins, ATP production. So basically the cytoplasmic content is going to increase, proteins increasing, RNA synthesis taking place, the protein which are synthesized, they are accumulating inside. So everything is just accumulated so that the cell can now enter S phase. What will happen in S phase? In S phase, the DNA is going to replicate. Replication of DNA. Why should DNA get replicated? Because at the end of this cycle, one cell is going to divide to give rise to two daughter cells, and both these daughter cells should get the complete information from the parent cell, and that is why the DNA replicates. One set goes to one daughter cell, another set goes to another daughter cell. So in this G1 phase, the cell has 2n number of chromosomes. It is diploid. DNA content is in 2n number of chromosomes, 2 sets. In S phase, the DNA, whatever is on 2n, replicates. That means here the DNA is doubled. It is equivalent to 4n. Complete DNA. That means in this case there are, if you are talking of human cell, 46 chromosome is the 2n number. Here it is 46 plus 46, because original 46 and after replication you have one more set. So the number DNA is doubled. So here replication of DNA content is doubled, and whatever is required for this, raw material and everything was prepared in G1 itself. So in S phase, no further accumulation of protein or synthesis of protein or all these things are not taking place here, only DNA related things. So DNA gets doubled. Coming to G2. After G2, the cell has to divide. So now it has to prepare itself for the division process. So in G2, the changes which would take place. Protein synthesis again. Why are proteins required here? Because these proteins will help in spindle fiber formation. These proteins would help in formation of newer membranes of the organelle. So this protein is different. Here also protein synthesis was taking place, but this protein was mainly histone protein, which was required for DNA replication. So protein types are also different depending upon which phase. We can write it here, example, histone. This is just one example, and in case of G2, the proteins which are required are for plasma membrane or spindle fiber. So protein synthesis is in both, but which type of protein is actually important, because this is the time when the cell is preparing itself for the division process. So protein synthesis. Certain organelle will have to get divided. That means half of the mitochondria would go into one cell, half would go into the other. If it is a plant cell, plastids would also replicate. New ER is to be formed. Other organelles are also required. So organelle either get duplicated or new organelles formed. ATP is anyways required. So ATP synthesis, because for division also energy requirement is very high. So ATP synthesis. Now the cell is ready for division. So after this it would enter the M phase. So during interphase what has happened is, first the cell prepared itself for S phase so that the DNA could be doubled. After S phase, in the gap 2, the cell prepares itself for the division, and now division will take place. How would the cell divide? This M phase or division phase will be completed in 2 steps. Karyokinesis and cytokinesis. In karyokinesis, the karyon or the nucleus will divide, followed by the division of cytoplasm. So once the cell divides, this was the original, we started with one cell. The cell went through G1, prepared itself, its DNA got doubled, it prepared itself for the division, and now the cell is dividing into 2 daughter cells. So one cell divides into 2 daughter cells. How many chromosomes would these 2 daughter cells have? Originally the cell had 2n number diploid. In S, those 46 chromosomes made a copy. G2, no change in the DNA, and here one cell gets 46, that is 2n, and other cell will also get 46, that is 2n. Now if this cell has to divide one more time, it will enter the next cell cycle, and if it has to mature and get differentiated, it will grow and then come out of G1 and will go into G0 phase. Here these are daughter cells, smaller in size. They will grow, accumulate enough cytoplasm, RNAs, proteins, organelle, everything, and will come out of the cell cycle. This is known as G0 phase. In G0 phase, the cell undergoes maturation and differentiation. Normally, the cells do not come back into the cycle, but there are certain cells which are capable of coming back into the cycle, like parenchyma cells. So parenchyma cells, if required, they are in G0, they are mature, they are specialized for particular function, but if they are required to undergo division, then they will come back into G1 and get into the cell cycle again and will divide. In this whole process, there are many changes which are taking place, and there should be something which is checking it. So in this cell cycle, there are 3 checkpoints. Checkpoints are basically areas or steps where checking is done. That means if everything is okay, then the cell will be allowed to move further. Otherwise, the process will stop there and the cell has to go back to the original. For example, let us take the checkpoints first, then we will see what all happens in these checkpoints. First checkpoint is towards the end of G1 phase. It is known as G1 checkpoint. It is the first checkpoint. It is somewhere here. This is G1 checkpoint. What is it going to check? It is going to check whether all preparation for S phase is done or not. That means, is the cell size okay? So when can we pass this checkpoint? You pass it if all these things are done, completed. Cell size is proper. If cell size is proper, the checkpoint is crossed. Protein accumulation, if protein accumulation for S phase is proper, all histone proteins and everything is synthesized, then the cell crosses this checkpoint. Is sufficient ATP synthesized? Then this checkpoint is crossed. So a cell starts from here, enters G1, towards the end there is a checkpoint. It is going to check whether the cell has done everything whatever is required. If yes, then the cell moves further, enters S phase. The second checkpoint comes towards the end of G2 phase. It is known as G2 checkpoint. So the next second checkpoint is G2 checkpoint. What is to be checked here? Up till now, what has happened after the first checkpoint? First checkpoint cross, that means everything is done. Cell size is proper, protein accumulation is proper, ATP accumulation is also proper, cell comes into S phase, DNA replication takes place, it comes into G2, more protein synthesized, everything done. What is being checked here? At G2 checkpoint, the points which are checked is DNA replication proper and complete? If yes, then this checkpoint can be crossed. Are all organelles properly divided? Number of organelle, is this number proper? Can proper number be given to two daughter cells? If yes, then this checkpoint will be crossed. Is sufficient protein ready for the cell to divide? So protein accumulation, yes, then this is passed. And last, ATP. So basically here also it is being checked for all the preparation. If this preparation is done, then G2 checkpoint is also crossed. Now comes the third checkpoint. Third checkpoint is in the M phase, and in the M or mitotic phase, because there is karyokinesis, and in karyokinesis there is prophase, metaphase, anaphase and telophase. So this is in metaphase of M phase. So it is also known as metaphase checkpoint. So third checkpoint is metaphase checkpoint. What is happening in this particular period is the division. So the cell has undergone prophase, metaphase. In metaphase, the chromosomes, they have to arrange on the spindle fiber, attached to the spindle fiber on the equatorial plane. We will be talking about the cell division after this section. So what happens is all the chromosomes align on the equatorial plane, and one side the spindle fiber attaches to it. Suppose this is the chromosome, and here we are talking about doubled DNA. So there is an original chromosome and a copy which was made in S phase. So spindle fibers would be attached to the centromere here and here. So is this attachment proper? If the attachment is proper, then only these chromosomes will go towards two sides, half the number here, half the number here. So totally if there are 4n, 2n will go towards one side and 2n to the other side. So we are talking about the two chromosomes getting separated during the M phase, that is metaphase, where the spindle fiber attachment is proper. If these spindle fibers are not properly attached, then the chromosomes will not get separated. So what is checked at the metaphase checkpoint is this one point. Are chromosomes attached to the spindle fibers? Is this attachment proper or not? And second, are all chromosomes on the equatorial plane? So all chromosomes on equatorial plane. If these two things are done, then it crosses the third checkpoint. So there has to be a checkpoint, otherwise there would be some kind of problem in the chromosome separation. And we know if chromosomes, if they are not properly separated, if both go together towards one side, all these things would result into mutations. Because every time cell goes through the cell cycle, it is giving rise to two daughter cells. Division of chromosome has to be proper. All the proteins which are required for the processes, they should be properly synthesized. If there is any mistake anywhere, then the two daughter cells which are formed, they would be faulty. Faulty in the sense they would have a different type of genetic material. That is known as mutation. So it could be chromosome number mutation. Some cell may have more, some cell may have less number of chromosomes. So these checkpoints basically ensure that the cell cycle runs in a proper manner. If a cell fails to cross a checkpoint, for example, if a cell fails to cross G2, what does it indicate? Basically, it indicates that something has gone wrong, either in G2 or in S. So either the cell will be discarded, that means removed from the cycle and destroyed, or it will restart from here. That means it will check whether the DNA replication is proper or not, all protein accumulation proper or not, and when it comes back to this place, again rechecking, and then it will be allowed to go further. So cell cycle and checkpoints. So these three checkpoints very important, which ensure that cell cycle works in a proper manner. Now let us take the different types of cell divisions.